Hello, my name is Emily Smith, and I will be introducing the COVID-19 project my biochemistry class has taken on. So, what does COVID-19 do and what is it? COVID-19 is a publicly known name for SARS-CoV-2, which means Severe Acute Respiratory Syndrome Coronavirus 2. This virus is a respiratory virus that not only attacks the lungs but other parts of the body, which makes this virus unlike other respiratory viruses that we have encountered up until this point. In this presentation, we will discuss the intricacies of this virus, so what it is, how it interacts and affects various parts of the body, and where we are at globally for any vaccines. COVID-19 is a virus that is spherical with projecting spike proteins off the spherical surface. It is these protein spikes that interact with ACE receptors on a human cell, which is when someone would then contract the virus. The COVID-19 virus contains RNA within the virus, which is used to make more copies, and proteins covering the outer membrane and the spike protein that protrudes the surface. The size of this virus is incredibly small. As you can see, I have a line drawn in the length of 100 centimeters, approximately the size of a yardstick. You can see that there is a basketball for reference at a diameter of approximately 24 centimeters. When zooming in, you can see the clip goes beyond the basketball and you see a 1 centimeter line drawn to show a size that would be most familiar to the public. Once we go beyond that 1 centimeter size, we reach a line that shows 20 micrometers. In comparison to this 20 micron line, we see two circles. The top one is the size of one human cell, approximately 10 microns, and an average respiratory droplet measuring at 5 microns. When we zoom in to look at the respiratory droplet, we see a small red mark to the right, and can zoom in to see the size of the virus, approximately 0.1 microns, so incredibly small. The virus itself is not stable enough to survive without being attached or transported by something else. Since the virus can't live by itself, we know that it can be transported through respiratory droplets. Though the virus is small, droplet size is too big to make it through the face mask fibers, thus stopping the spread of the virus. When looking at appropriate face masks for prevention of COVID-19, the best mask the CDC or Center for Disease Control recommends is to use the N95 face mask. To make a comparison, it would be like trying to fit a beach ball that is 18.75 feet in diameter through a golf ball hole, which is 4.5 inches in diameter. Focusing on the human cell, we can see the same virus size at 0.1 microns, but it's twice as small in comparison to the human cell than the respiratory droplet. With the virus size at 0.1 microns, the spike protein that interacts with the human cell measures only at 0.01 microns, 1/10 the length of the virus diameter.
Hi, I'm Lena and I'm going to be continuing the story.
Coronaviruses are a diverse family of viruses. They have a spiky protein surface reminiscent of a crown when looked at under electron micrographs, as seen here. The spindles you see coming off the virus are known as spike proteins. Here is an enlarged version of a COVID spike protein. With this spike, you see the top and outer fragment, colored gold, which binds to cellular receptors, while the inner fragment, colored orange, directs fusion of the virus to the cell. The surface of the virus is shown in silver. Many other viruses over the years have used similar spiked proteins to infect cells. The virus most commonly compared to COVID-19 is the SARS-CoV of 2003, shown on the right. The SARS virus was more deadly but less easily transmissible than COVID-19. As you can see, the number of COVID-19 cases is in the millions, while SARS is in the thousands. However, its death rate is only 2.3% compared to the 9.2% of SARS. So why is this? A possible explanation for this is the greater amount of virus present in the nose and throat much earlier in the contraction of COVID-19. This is to say that it carries a greater viral load. Additionally, COVID-19 can be spread without symptoms, making it difficult to track. Both the structures of SARS and COVID-19 are extremely flexible, which gives them a great advantage. Due to their flexibility, they can more easily adapt to bind to a given receptor. You can see the similarities between the structures of these spikes, though varying rates of flexibility and size can affect how dangerous and transmissible each virus is. The colors I chose as I generated these images is reflective of their properties. For example, the most dangerous part of COVID-19 is the recognition section, colored here in an alarming gold. For SARS, the most dangerous part is the fusion section, colored here in a deep maroon. Influenza is another dangerous spike protein, and it was responsible for the deadly 1918 flu. The 1918 flu, also known as the Spanish flu, was estimated to have 500 million cases and a 2.5% mortality rate. It was extremely virulent and only exacerbated by the recent war of that time, resulting in malnourishment, overcrowding, and poor hygiene. The structure of influenza hides the fusion end well, making it difficult to target with vaccines. Additionally, the recognition section is extensive and constantly mutating. As you can see, the recognition section extends much farther down on this spike protein. It's shown in peach.
Hello, I'm Lucas Downen, and I will be taking over where Lena had left off. And I am going to be covering the spike protein and the sugar trees. And now it is time to talk about the spike protein. The part of the protein that binds on the spike protein is most often buried inside the molecule and only comes out when binding to host cells. In this case, the ACE2 receptor. This prevents the immune system from recognizing and binding to the structure. This also poses the same issue when developing the vaccine. The spike protein consists of an extracellular N-terminus, a transmembrane domain, which is anchored in the viral membrane, and a short intracellular C-terminal segment. The SARS-CoV-2 largely consists of a signal peptide located at the N-terminus and the S1 and the S2 subunits. In the S1 subunit, there is an N-terminal domain and a receptor binding domain that recognizes and binds to the host receptor. The binding of virus particles to cell receptors on the surface of the host cell is the initiation of virus infection. Therefore, receptor recognition is an important determinant of viral entry and a drug design target. The S2 subunit houses the fusion peptide, heptapeptide repeat sequence 1, also known as HR1, as well as HR2, the transmembrane domain, and the cytoplasm domain. SR2 generally mediates viral cell membrane fusion by forming a 6 helical bundle. In the native state, the COVID spike protein exists in a pre-fusion conformation. During viral infection, target cell proteases activate the S protein by cleaving it into the S1 and the S2 subunits, which is necessary for activating the membrane fusion domain after viral entry into the target cell. Once the virus interacts with the host cell, extensive structural rearrangement of the spike protein occurs, allowing the virus to fuse with the host cell membrane. The spikes are coated with sugar molecules to camouflage them, evading surveillance of the host immune system during entry. RBD binds to the ACE2 receptor, and the S2 subunit changes conformation by inserting the fusion peptide into the target cell membrane, which then exposes the HR1 domain and triggers interaction between the HR2 and the HR1 domains, thus bringing the viral envelope and cell membrane into proximity for viral fusion and entry. Let's talk about the significance of sugar trees when it comes to the spike protein. When a virus infects a human cell, the cell's ribosome picks up viral RNA and translates it into proteins inside of the cell. Cellular enzymes start adding mannose sugars to newly synthesized proteins as they exit the ribosome. Once synthesized, these proteins are then excreted into the cell, and along the way, the sugars are maturated and trimmed back so that the sugar structures have the appearance of those that are normally made by the host cell and are also more complex and branched in nature. In the image provided, you can see that the COVID spike 2 protein is pretty heavily covered in sugars. The color scale shows the percentage of these sugars at various sites along the protein. The higher the content of the sugar, the less they resemble those of the host cell, and the less camouflaged the protein is from a host's immune system. The ACE2 binding site is the portion of the protein that interacts with these enzymes on the cell's surface. The S1 and S2 refers to the two individual parts of the subunits that we spoke about a little bit ago. As we just said, these sugars don't just camouflage the virus from the host's immune system as sugars normally do, but they also help the spike protein take on the conformation that equips it to infect cells, as well as helping the protein fold. In order to infect a cell, the spike protein must undergo the conformations which we spoke of earlier, where its receptor binding domain must raise itself upwards toward the host cell receptor to make this connection. These two sugars appear to tuck themselves under this domain and help lift it toward the receptor in order to infect the cell. These sugars are also known to camouflage themselves like the proteins of the animal or person that has been infected. Viruses are known for commandeering the enzymatic machinery that host cells use to add sugars to their own proteins and get those enzymes to add the viral sugars instead. But so why is it important to talk about the sugar trees? Well, for the spike protein, it's able to become more stable. As a result of this, it provides a lot more variation in how it can cover the protein, and these sugars make it harder to generate antibodies against them. These sugar trees love water, and this allows them to protect the protein and also become much stickier, making it easier for the virus to bind to respiratory droplets. These new sugars look very familiar to the body's immune system and are less likely to be attacked. It is important to find regions when not masked when it comes to looking for a vaccine.
My name is Ashley Cookett, and I will be reviewing how the coronavirus attaches to cells. To begin, I will pick up on the spike protein where Lucas left off.
Shown here is the spike glycoprotein of the coronavirus. To orient the picture, this area here would be the virus capsule, and down at the end in orange is the receptor binding domain that will attach to a cell. These binding domains are currently in the closed conformation, and as we can see in the right picture, there are 3 separate binding domains. As the virus is inside a body and preparing to attach to a cell, one of the receptor binding domains will hinge open, and this will allow for better attachment to the receptor.
The receptor that the coronavirus attaches to is the angiotensin-converting enzyme 2, or ACE2, as shown here. This enzyme sits within the plasma membrane of the cell. When looking at the image, we can imagine the plasma membrane extending on either side. This means that this side is extracellular, where the virus will attach, and this side faces the inside of the cell. This is what the spike glycoprotein looks like as it is attached to one ACE2. Once one ACE2 is bound to the spike protein, a second receptor binding domain will open and attach to an ACE2, followed by a third. This image shows the spike protein fully bound to the cell membrane via ACE2. We can see that the receptor binding domains are all in the open conformation. The glycoprotein attaches to 3 ACE2 to form crosslinks. As shown here, we can see how tightly the receptor binding domain is to the ACE2. The close proximity tells us that they are tightly bound together. These final images show the same bonds with different representations. The image on the left shows the backbone of the spike protein and the ACE2. Then the image on the right shows the secondary structures and color and a translucent shell to depict how much space the molecules take up.
Hi everyone, my name is Emma Liddicoat, and I will be stemming from the work previously mentioned by Ashley to further discuss the ACE2 receptor and how it is connected to our human bodies. I am first going to touch on exactly what the ACE2 receptor is, and then move on to explain its role and discuss where it can be found in our bodies. Let us first focus on the image presented. Angiotensin converting enzyme 2, or ACE2, as it is sometimes called, depicted here in blue, is a membrane-associated protein that acts as a cellular entry receptor for SARS coronavirus 2. It is a specific protein that helps the virus infect human cells. In fact, scientists have demonstrated that SARS coronavirus 2 does not use any other receptors associated with the coronavirus family. Angiotensin converting enzyme 2 provides a direct entry point for SARS coronavirus 2's spike protein, which is depicted here in orange, to bind to it like a key being inserted into a lock. The protein, angiotensin converting enzyme 2, is found on the surfaces of many cell types and in many tissues throughout the human body. Now let's focus on the provided image. Here is a depiction of the viral entry and replication process of SARS coronavirus 2 into a human cell. Starting at the left side of the image, upon viral entry into the cell, the virus is broken open and its messenger RNA, or its genetic material here, is released into the cell. The virus's genetic material is replicated, packaged, and finally assembled for the production of multiple copies of itself. The multiple copies leave the human cell via a process called budding and are released throughout the rest of the body. With the virus in the body, we now shift our focus to the ACE2 receptor. The major role of angiotensin converting enzyme 2 in the human body is to convert angiotensin 2 to angiotensin. This means that not only is ACE2 a receptor for SARS coronavirus 2, but that it also regulates many physiological systems within the body. One such system is the renin-angiotensin-aldosterone system. This system plays a key role in regulating blood volume and systematic vascular resistance. The elevation of blood volume and arterial tone in chronic health circumstances is what this system is a critical regulator of. This system regulates these such bodily functions by either increasing sodium reabsorption, water reabsorption, and/or vascular tone. The mechanism of the renin-angiotensin-aldosterone system begins with the formation of renin, which acts on its target, which is angiotensinogen. Angiotensinogen is produced in the liver and is found circulating the plasma. Angiotensinogen is cleaved by renin into angiotensin 1, which we can see here depicted in the first circle. Angiotensin 1 is a precursor for angiotensin 2, here in the bottom circle. Angiotensin 2, if we follow this arrow going down this direction, functions to increase blood pressure and inflammation, which increases damage to blood vessels and different types of tissues. Angiotensin converting enzyme 2, now following this arrow going to the left, converts angiotensin 2 into other molecules that help counteract angiotensin 2's effects. Angiotensin converting enzyme 2 is a critical element in the overall biochemical pathway. In relation to SARS coronavirus 2, angiotensin 2 increases inflammation and also the death of cells in the alveoli, which are found in the lungs. The alveoli are responsible for the uptake of oxygen into the body. Thus, the effects of angiotensin 2 are harmful but reduced by ACE2. The binding of SARS coronavirus 2 to a human ACE2 receptor, now we're following this smaller arrow right here, prevents ACE2 from its normal functions, which are regulating the effects of angiotensin 2. So with the regulating mechanism of ACE2 inhibited by the virus, more and more angiotensin 2 is able to be made. The ability for angiotensin 2 to be produced enables it to be able to cause injury to tissues throughout the body. The increase in angiotensin production is plausible to the contribution of injury to the lungs and heart of COVID-19 patients. Understanding not only the function of the human ACE2 receptor, but the location of it as well has played a large role in COVID-19 research. The most commonly known area in the body that ACE2 proteins are found in is the lungs. Other areas of the body include the heart, blood vessels, kidneys, liver, and gastrointestinal tract. The protein is also found in epithelia cells, which line certain tissues throughout the body and create protective barriers. Cardiac, lung, liver, and renal organ systems have a higher rate of expression of ACE2 receptors than other body systems. These bodily systems are thus more susceptible to higher hyperinflammation and damage. Individuals with previous health conditions such as diabetes, high blood pressure, metabolic syndrome, and those with advanced age tend to express organ systems with lower numbers of ACE2 receptors, leaving them prone to higher fatality rates. The human body is incapable of making its own antibodies for SARS coronavirus 2 and thus must be infected with a virus to allow the body to produce the needed antibodies and fight off the infection. Of course, becoming infected with the virus comes with many risks and is why there is such a high demand in research for the formation and distribution of a safe and usable vaccine.
Hello, my name is Bryn Schubert, and I will be adding on to the spike protein portion that Lena previously explained, as well as introducing the topic of antibodies. This section will focus on immunology and the science behind the vaccine. In order to understand how the vaccine and treatment for COVID-19 work, we need to understand what antibodies are. Antibodies are our body's way of recognizing foreign substances that make their way into our bodies. They attach using their flexible arms, which can be seen here and here, flagging the foreign substance for the body's immune system to then come and attack the substance. The antibodies have common arms with two heavy and light chains. The light chain can be seen as the teal turquoise color, and the heavy chain can be seen as the bluish violet color. The hinge region of the antibody, seen right here, can be seen between the two arms, allowing for flexibility and specific attachment onto the foreign substances. These antibodies are from the actual COVID-19 patients and will recognize and attach to the spike protein of COVID-19. Part of the treatment for patients diagnosed with COVID-19 is using serum from other COVID-19 positive patients who have developed antibodies that recognize COVID-19. These antibodies are collected through plasma donation and are transfused into other positive patients. The antibodies that are specified to recognize the spike protein on the COVID-19 viral surface are then able to latch onto the spike protein using its heavy and light chain arms, signaling for the immune system to start its response in neutralizing the virus. see the COVID-19 spike protein in the red. The antibodies we see are shown as the backbone model. We can see the 3D image, and then we can also see the backbone model behind a translucent 3D image. Both of the recently developed Pfizer and Moderna vaccines were developed by using mRNA strands of part of the spike protein to introduce to the body. This allows our own cells to make many copies of the spike protein so that our bodies can recognize and build an immune response. This is caused by antibody development by the body's immune system against the COVID-19